Hello everyone. Our next topic is blast and substitution matrix. Okay, so let's begin with the class. So, um, what is BLAST? So, BLAST is a sequence similarity search program for comparing biological sequences such as the amino acid sequence, or you can say like nucleotide sequences of different protein of different nucleotide of DNA sequences with sequence database or library sequences. Okay, so what is doing BLAST is, suppose you are having a query sequence. Okay, so what query sequence can be either a protein or can be a DNA. Okay, so it will search against any kind of sequence databases, like if you are having protein sequence, it will search against the protein database or the nucleotide database, or if you are having the nucleotide sequence, suppose you are having here protein sequence, then it can search either nucleotide database or can by protein sequence. If you are having nucleotide sequence as a query sequence, then it will search again the protein sequence or by the nucleotide sequence. Okay, in either way it can perform the search. Okay, so what is BLAST? BLAST is B stands for basic, L stand for local, A stand for alignment, and S stand for search, and T for tool. So basic, local, alignment, search tool. Okay, so it will identify, or it will predict the similar matches against the databases. Whatever databases you will provide as a query, it will search against those databases. Okay, so in the BLAST there are different kind of BLAST, basically amino acid sequence BLAST or DNA sequence BLAST. Okay, it is categorized into two different type of BLAST. First one is the amino acid sequence BLAST and DNA sequence BLAST. Okay, so in amino acid sequence BLAST there is two type of BLAST. First one is the BLAST P and second was the T BLAST N. Okay, in DNA sequence there is BLAST N, BLAST X and T BLAST X. Okay, so here we will see, like in this column many questions have been asked in previous year questions, so we will see this column very carefully. Okay, so if you will see the first one, that is the BLAST N. Okay, so the program name is BLAST N. Okay, it is a kind of nucleotide or DNA sequence BLAST. Okay, here you are giving a query sequence as a nucleotide sequence. Okay, so there are two boxes there in the BLAST program. Okay, two boxes there. So one is your query sequence you have to provide. Okay, as a input in FASTA sequence you have to either provide the nucleotide sequence or you have to provide the protein sequence. Okay, so this will be your target sequence. Okay, so against which this BLAST can be performed. Okay, so the first one is the BLAST N. So in BLAST N what will be happening here, the query sequence is nucleotide. Okay, this nucleotide either can be DNA or can be RNA. Okay, it will search against what kind of database? So it will search against the nucleotide database. Okay, in the previous slide I have told you earlier that the nucleotide database can be either searched against the protein database or nucleotide database itself. Like that, protein query sequence also searched against the nucleotide database or protein sequence. Okay, so here the program is BLAST N. So BLAST N query sequence is nucleotide and database is the also nucleotide. Okay, the second one is the BLAST P. Okay, this is the simplest form of the BLAST. Okay, so BLAST P, here also query sequence is protein and database is also protein. Okay, so here in case of BLAST N both the thing are nucleotide. In case of BLAST P both the thing are protein. Okay, things are protein. So here if you can if you want to remember, like N denoted nucleotide, so both the thing is nucleotide. P denoted to protein, so here both the thing is protein. Okay, so then you are having mixed blast. Okay, so mixed blast, the first one is the blast X. Okay, so blast X, how you will remember, like blast X is you are having the nucleotide sequence, but you wanted to search against the protein sequence. Okay, so in this case the query sequence will be your nucleotide. So this program, what it will do is it will convert the nucleotide into protein sequence by considering all the 6 ORF. Okay, reading frames. Okay, open reading frames, it will consider all the 6 open reading frames, and then it will translate into the protein, and this protein sequence again search against the protein database. Okay, so here your query sequence is nucleotide, but you are searching against the protein database. Okay, that is that thing is done by blast X. Okay, the next one is the T blast N. So T blast N is just opposite of this blast X. Okay, so T blast N, what it is doing is, T blast N basically here you are having the protein. Okay, query sequence is your protein. Okay, so and here you are having database is translated nucleotide. Okay, so here you are searching query sequence as a protein, and against whom you are searching the translated nucleotide. Okay, so it will again translated nucleotide means it will again means it will be like protein sequence only. Okay, but the databases which is having the translated nucleotide. Okay, it will perform the blast against the translated nucleotide. T blast X is both the things are translated. Okay, and both the things are nucleotide. That you have to remember. T blast X is both the thing is translated and both the thing is nucleotide. Query sequence is also nucleotide and the databases is also nucleotide. Okay, that you have to remember. So how you will remember these things? So first one is the blast N, simply N denote for nucleotide, so both the thing is nucleotide. Okay, so blast P is basically P stand for protein, so both the thing here is protein. Okay, blast X is X is here translated nucleotide. Okay, so when you are having blast X, that time what is happening, the translated nucleotide is searched against the protein database. Okay, so that nucleotide is firstly translated into protein, then protein query sequence is searched against the protein databases. Okay, T blast N means here you are having the protein sequence and you are searching against the translated nucleotide. Okay, that will be also protein. Okay, so but it is present in the form of translated nucleotide. Okay, then you are having T blast X. T blast X is translated nucleotide search against the translated nucleotide. Okay, translated nucleotide, both the thing are the translated and both the thing is nucleotide. Okay, so this is the different in blast. There is two kind of substitution matrix which is used to score the aligned position in a sequence alignment procedure. Okay, it can be amino acid sequence or it can be nucleotide sequence. Okay, so what it means, suppose you are aligning two sequences. Okay, so when you find a match or mismatch, that time to giving score you are either use the you use substitution matrices. Okay, so there are two common substitution matrices. One is the PAM. Okay, one is the Blossom. Okay, one is the PAM and one is the BLOSSOM. So PAM is nothing but point accepted mutation, and BLOSSOM is block substitution matrix. Okay, block substitution matrix. So this is the different kind of PAM matrices, and this is the different kind of BLOSSOM matrices. Okay, so what when we should use the PAM matrix and when we use the BLOSSOM matrix? So when you are comparing closely related sequences, okay, you know that two sequences are closely, but how much they are close, that time you need to use the PAM matrix. Okay, so lower the PAM suppose your two sequences very much identical to each other. Okay, so you have to use the PAM 1. Okay, it means it is having 99% identity. Okay, and only 1% divergence. Okay, and suppose when you wanted to, yeah, so to compare closely related sequence, lower number of PAM matrix you have to use, and to compare closely related sequence, higher number of BLOSSOM sequence you have to use. Okay, higher number of BLOSSOM you have to use. Like that, when you want to compare distantly related protein, that time you require higher PAM values, and BLOSSOM lower BLOSSOM values. Okay, so here if you will see here in this graph, this is common or medium PAM and BLOSSOM values, that is PAM 120 and BLOSSOM 62. BLOSSOM 62 is default substitution matrix for blast alignment. Okay, so that you have to remember, like BLOSSOM 62 is common default substitution matrix in blast. Okay, so when you go for lower PAM matrix like PAM 1 or higher BLOSSOM matrix, that is the BLOSSOM 80 or more than 80, that means your sequence is less divergent or they are very close. Okay, they are very close to each other, very close to each other. Here both is having equal things, like whatever thing done by PAM 120, that is BLOSSOM 62 is done the same thing. Okay, so there is no like similarity or identity kind of thing. But when you go from higher PAM or lower BLOSSOM thing, it means like higher PAM 250, BLOSSOM 45, it is less value. Okay, it means your sequence is more distant. Okay, your sequence is more distance. Your sequence is more distant, you have to remember. Okay, so in case of very close sequence, you have to use low PAM value and higher BLOSSOM value. And in case of more distant protein alignment, you have to use higher PAM value and low BLOSSOM value. In this question, it has been asked like which one of the following tool is used to compare all the possible 6 open reading frames of a given nucleotide query sequence with all the available 6 open reading frame of the nucleotide sequence databases. Okay, so here it is asking like we have to the query sequence is here also the nucleotide sequence. Okay, nucleotide query sequence, and the matching sequence should be also the nucleotide means the database also should be matched against the nucleotide sequence databases. Okay, so here both the thing is nucleotide. Okay, so you are like matching the nucleotide sequence with the nucleotide databases. Okay, so but here it is saying all the possible 6 open reading frames. Okay, so we have no we have seen that here in case of the T-BLAST X, okay, which compare the nucleotide query against translated nucleotide sequence databases. Okay, here all the 6 nucleotide query sequence, all the 6 nucleotide query against the translated nucleotide sequences databases, it will be like checked. Okay, so in case of the blast X, it is 6 frame conceptual translation product of a nucleotide query sequence against the protein sequence databases. Okay, here the protein sequence is databases is used to like do the blast. Okay, but here the query sequence will be nucleotide, as well as it is means checked against the translated nucleotide sequences. Okay, so that's why yeah, so that's why the correct answer of this question will be option D, T blast X. Okay, because in case of T blast N, the the nucleotide sequence 6 frame translated product of a nucleotide query, okay, checked against the protein sequence databases. But in question it is asking about the query sequence will be in nucleotide database, nucleotide sequence, and it will be checked against the nucleotide sequence databases. Okay, so that's why the correct option will be T blast X, whereas this blast N is compare the nucleotide query against the nucleotide sequence databases. Okay, but here the translated thing is not there. Okay, but in the question it is asking about the translated thing, right? Because it is questioning about the compare all the possible 6 open reading frame. Okay, so that's why here blast N will be not the correct answer. Okay, because here it is not comparing against all the 6 open reading frame. Okay, so that's why it is not the correct option. And the blast X is nothing but it compared the 6 as I earlier told you, it will check the 6 frame conceptual translation product of nucleotide query sequence against the protein sequence databases, whereas this T blast N compare protein query, T blast N will compare the protein query against the 6 reading frame of a translated nucleotide data sequence databases. Okay, it is the just opposite of T blast X. Okay, in case of T blast N, there is the translated nucleotide sequence databases, and in case of T blast X, there is the translated nucleotide sequence databases. Okay, so the query and here the query sequence is protein query sequence, and here the query sequence is the nucleotide query sequence. Okay, so that's why the correct option will be option D, because here it is asking about the nucleotide query search against the nucleotide sequence databases. Okay, 6 open reading frame, that you have to remember, otherwise it will be normal blast N if it is asking about the nucleotide query sequence search against the nucleotide sequence databases, then it will be blast N. But it is asking about the 6 open reading frame of a given nucleotide to the 6 open reading frame of a nucleotide sequence databases. Okay, so that's why the correct option will be